Hello, my name is Mr. Chipman. This is 4.2 Introduction to Signal Transduction. I teach biology and AP Biology in Murray, Kentucky. And I teach a lot of you AP Biology. This is kind of cool. It's been great to receive messages over the years, even the last year, just how this channel is helping you out. If you think the same, leave me a comment. I just love to read those. I hang them up in my classroom even. They're very encouraging. Let's look at 4.2. So this idea of signal transduction is very important for this unit. In fact, the whole unit sort of revolves around this. Even the cell cycle unit, or the portion of the unit, really involves this sort of idea as well. Signal transduction involves three stages: reception, transduction, and response. Really, transduction is the only one that sort of needs a little bit of a fleshing out. Reception is the reception of a signal molecule. Transduction is any sort of relay pathway that gets the signal to the response, right? Sometimes relay pathways are going to be really simple. Sometimes relay pathways can be quite elaborate. And then response is going to be some kind of cellular response. A lot of times the cellular response is going to be make a protein. Right, but the cellular response doesn't necessarily have to be make a protein. Muscle contraction is a great example of a not make a protein response. And so, but there are a lot of times we're going to see the response happen on the DNA. The DNA is going to trigger protein synthesis and so forth. And so very important signal transduction, these three steps, you will see them a lot in this unit. In fact, very important even for the next couple of units, just understanding overall cellular processes. A couple of key components that we need to understand when it comes to signal transduction. One of these is the word phosphorylation. We've talked about phosphorylation before. Just to remember, phosphorylation is when a phosphate is added to something. Usually when something is phosphorylated, it is turned on. Right? It is a process that is being turned on, kind of like going and flipping a light switch, adding that phosphate to something. ADP or ATP coming and putting a phosphate on something typically activates whatever that something is. It's usually a protein. A lot of times you will see the term kinase associated with activation as well. A kinase is a protein that turns on another protein, whereas a phosphatase is one that usually removes a phosphate from that, sort of switching that protein off, right? And you see this in this cycle of a protein being turned on and one being turned off through the use of kinases and phosphatases, that phosphorylation. Very important idea. Kinase and phosphatase are words that you're going to see a lot in this unit, and just understanding that when you see them, they mean a certain thing is very, very good. All right, then the idea of amplification. I think we all understand the idea of amplification, right, where something that's small gets made to be larger, right, through any number of things that can happen. We think about sound amplification, for instance, like a speaker or something like that. And so in this particular case, we have signal amplification. This is where one signal molecule can end up eliciting dozens or hundreds of responses because of a signal cascade, right, where one molecule can start two processes, and those two processes can start two more each, and you get the idea, right? And eventually you just get whoosh, you get really big. And what is the idea behind that? The idea behind that is resource management, right? Saving resources, saving energy, and all of those different processes can sort of be scaled in that way. And some processes need amplification, like growth, emergency response, anything like that that requires a lot of response all at once. Amplification's a good thing. I used to think about this as like a phone tree, but phone trees are kind of antiquated now, since things like group messages and email. They've been antiquated since like the 90s, but here we are. All right, let's talk about reception a little bit. We've mentioned this a little bit in the previous section, but it bears some reminders here. A ligand is a signal molecule, and the receptor is usually specific to the ligand. I say usually. There may be cases where it's not, but it's very similar to sort of the enzyme substrate idea, right, where the enzyme is specific to the substrate, and the receptor is going to be specific to the ligand in this case. And that is a very important idea in this, because a target cell is going to have receptors on it that are going to match the sending molecule. Very important idea. Receptors are typically proteins. Proteins have their shape. No shape determines their function. We know all of this from previous units, and this is going to continue to be a theme that we're going to talk about all year long. Signal molecules, and I don't have a picture for this, that's why I kind of had to drop back and punt there. They can either be on the membrane, so you have a molecule can be on the membrane, the receptor can be, or it can be inside the membrane. You will see an intracellular receptor as well. Some ligands are able to pass directly through the cell membrane. Why would it be able to do that? Because, well, it's nonpolar. Like a steroid sometimes can pass directly through the cell membrane and match with a receptor inside the cell. Still following this process, reception, transduction, response, but sometimes the reception happens on the cell membrane, sometimes it happens within the cell, depending on the chemical makeup of the ligand. Let's look at this idea of transduction. Transduction is, as you can tell, can be very complicated, and so we need to spend some time on here. And so a couple terms that you need to be familiar with. Actually, I'm going to go back up here. When reception occurs, there elicits something called a conformation change, right? When that ligand binds with the receptor, it causes the receptor to change shape, right? And think of it as, it's like a typical or a very temporary kind of chemical reaction, right? Where one chemical comes in contact with another one, and because they do that, they change shapes, right? And this shape change is going to cause this cascade to begin to happen. And so when you have this ligand bind to blue molecule up here, some kind of receptor, that receptor is going to change shape. And that shape change is going to start this whole kind of pathway. And this pathway, all the way from receptor changes to cellular response, all of this inner workings here is called transduction. Transduction is basically that signal being relayed from reception to cellular response. And there's going to be a series of shape changes that are going to occur here. And a lot of times what the difficulty is you're not going to have to memorize a particular pathway, right? This particular pathway looks like some sort of muscle contraction pathway, right? You don't have to memorize that pathway, but you just need to be able to look at a diagram like this and sort of understand what's going on, right? We can see that ligand binds to blue molecule, and blue molecule then signals or turns on phospholipase C, which is then going to elicit something called DAG, and then that's going to turn on IP3 and also protein kinase C, right? Protein kinase C is going to phosphorylate its targets, whatever its targets are, and then that's going to lead to some cellular response. IP3 is going to go into the ER, and that is going to bind with that protein channel on the ER, which is going to cause calcium to be released into the cytoplasm, and the calcium is going to bind to proteins, and the calcium binding to those proteins then elicits some kind of cellular response. You don't have to memorize any of that, what I just said. I was just looking at the picture and sort of just working through it, right? And so I strongly recommend that when you see a picture like this, that you kind of make that mental thing. A lot of times when you see a picture like this, it's either going to be on an FRQ, right? So you have several questions you have to answer, or it's going to be the multiple choice type of question where it's like for the next 5 questions, use this passage. And so it's going to pay out some long-term benefit for you to sort of walk through that picture mentally and maybe even make little notes on it if you're able to do that. If you have a paper test, strongly recommend that kind of work. A couple of terms that you need to have here that I don't have pictures for. One of those is secondary messengers. A secondary messenger is just a relay molecule, right? It's just a secondary messenger. It's like in between the primary and wherever it's going, right? A lot of times secondary messengers play important roles in signal amplification. Let's see if I can see one here. Yep, you can see cyclic AMP is one that you'll see a lot of times. And so response is how the cell is going to respond to the signal molecule that it received. All right, there's some, the sending molecule obviously had some intent behind sending that signal, which probably came from the brain. There's a whole thing that can happen there. But that response is going to be either one of the few following things. It could be gene expression. Gene expression is just a fancy word for making a protein. DNA codes for proteins. That gene is expressed when a protein is made. You get that. It could be cell growth, could be cell division, could also be elicited, like that. That signal response is going to cause a cell to divide or grow, like if there's an injury or something like that, or repair, anything like that. Any sort of metabolic change, like fever is a great example. A fever is something that is where the body increases metabolism on purpose, right? To increase body temperature. That's going to be brought on by some kind of signal molecule. There's literally just many that you could talk about here for response. But the idea is that the response is at the end of the pathway. You'll see it here. A lot of times you'll see this is gene expression, right? Cell growth, proliferation, survival, all part of the ERK signal transduction pathway, of which I have no idea what any of this stuff stands for, right? But you can sort of walk through and at least understand the pathway without knowing what SOS and RAS and any of that stuff is. Let's look at some examples. The main example to talk about here is the G protein coupled receptors, G protein pathway. G protein is a very common messaging pathway in a lot of living things. It's one of those conserved processes that we talk about. G protein is found in a lot of living things. And so this is where you have the G protein is right here. You see that. It is like a transduction type molecule. You have the signal molecule that comes in, receiver or the receptor receives it, and then it triggers this G protein to turn on, which is going to go turn on some enzyme, which is going to turn on some secondary messenger, which is going to elicit some kind of cellular response. G protein, very commonly used pathway. Over here, this picture here, you see an intracellular receptor, right? Some kind of signal molecule is able to pass directly through the plasma membrane, probably because it's nonpolar, and it is received inside the cell, and then that response is elicited. Let's see if I've got some more. I do. Ligand-gated channels. Ligand, reception molecule, gated. There's something that is closed that could be opened. How is it opened? Through a ligand. And it's an ion channel, so it allows ions in. See, the ligand receives the receptor receives the messenger that causes the gate to be open, and the little ping pong balls fly in. And then when the messenger or the ligand is removed, the receptor or the gate closes and causes those ping pong balls to be trapped outside the cell. Pretty neat. This is a lot of time. This is how neurotransmitters generally work, is this way, but there are other things that work this way as well. Let's look at a practice problem. A cell is exposed to a hormone that binds to a G protein coupled receptor. We just learned about that, right? On its surface, researchers use inhibitors to block each step of the signal pathway and measure downstream gene expression. Which of the following experimental outcomes best supports the claim that CAMP functions as a secondary messenger in this pathway? CAMP is cyclic AMP, but you don't need to know that. It could be called George, or it could be called triangle. Doesn't really matter. Which of the following experimental outcomes best supports the claim that CAMP functions as a secondary messenger? We know that a secondary messenger is just a mid-pathway sort of point, right? It's a relay molecule. And so what are we looking for here? They use inhibitors to block each step, meaning if they blocked cyclic AMP, then you'd expect gene expression to be blocked as well. Let's go see if we can find that in ABCD. When CAMP production is inhibited, the hormone still binds to the receptor, but the target gene is not expressed. Wow. Right there on A. This just, again, this underlies the idea that I'm going to go ahead and I'm going to look for an answer, right? I have formulated the response in my head before I start reading those responses, and that is what I'm looking for, right? And man, it just hit the nail on the head right here. Now let's look at BCD just to make sure, but I'm pretty confident with A. When a hormone is prevented from binding to the receptor, CAMP production increases. It wouldn't do that, right? It wouldn't do that. When a receptor is internalized in the cell, CAMP production increases and transcription. There's no reason to believe that, right? And it has nothing to do with the prompt either, about blocking or anything like that. And again, we're asked to experimental outcomes here. C has nothing to do with anything. When the gene has already been expressed, additional hormone decreases. Again, this doesn't have anything to do with downstream gene expression and the experimental that's going on here. A is by far and away the best answer. I really hope this was helpful. 4.2 unlocks this entire unit, I think. And so spend some time with it. We're going to be moving on to 4.3.